The CAR-T cells is the patient autologous T lymphocytes, what we build in a special receptor inside of these T cells, so they get specialized against one antigen. And there are different type of CAR-T cells and different type of transduction as we can build in this special receptor into the T cells. I work on especially called the CART-meso T cells. And our work right now, what we do, we use a self-inactivating lentiviral vector to transfer the meso-CAR inside of the autologous T lymphocytes of the patient.
And why did you choose mesothelin as an adjunct or a conjugate with the CAR-T cell?
Because mesothelin is not expressed in most of the normal human tissues, but expressed in a small group of human malignancies. And these are the malignant epithelial mesothelioma, the epithelial ovarian carcinoma, and the pancreatic ductal adenocarcinomas.
So the clinical objective would be what?
Would be what? That the T cells are identifying the mesothelin expression on the surface of these cancer cells, and they attack these cancer cells and kill them.
So the T cells are able to do the killing, and you can help them to find the cancer.
Indeed, they are serial killers. So a T cell can kill 1,000 to 2,000 cancer cells if they are able to, and we help them to identify who have to be killed.
Mm-hmm. You've been conducting a study then. What did you do?
So I'm conducting the study with two other physicians together, for Dr. Andy Haas and Dr. Gregory Betty. So three of us together are focusing in this free group of cancers. So after the patients enrolled, the patients undergo an apheresis, and we collect the T lymphocytes. And the T lymphocytes sent to a laboratory where they manufacture the CART-meso T cells. And the CART-meso T cells are ready. We infuse these CART-meso T cells into the patient's circulations, and we follow the patients, evaluating the safety, the feasibility of the CART-meso T cells, and also the clinical efficacy.
And how many patients have you treated with which sorts of cancers, and what happened?
So in this, today I represented the first cohort, which is 6 patients. 2 patients with a recurrent pancreatic adenocarcinoma, both of them stage 4, 2 patients with epithelial ovarian carcinoma stage 3C and 1 stage 4, and 2 malignant epithelial mesothelioma stage 3 and stage 4. And it was exciting because after 28 days, we saw that 4 patients developed stable disease from the 6 treated patients, so the disease stabilization rate was 66% in the first cohort. And this is just the start.
That is exciting because you took patients whose outlook was not very good.
These particular patients are heavily pre-treated, and they usually already consumed all or most of the standard of care options. And to reach a 66% disease stabilization rate amongst those patients who have progressive disease, it's a great success.
So why is it that your CART-meso cells are so good?
Why is it? Because these are very specialized on these mesothelin molecules. So when we transfer this meso-CART into the T cells, the CART redirect the T cells to identify the intact mesothelin molecule on the tumor cell surface. And after, as they identify, the CART therapy takes advantage of the cytotoxic potential of the T lymphocytes to identify and kill these tumor cells.
Is there a hope of quite an extended remission from disease?
I think it is. This particular meso contains most sequences, so it persists in the patient's blood about 3 to 4 weeks. But we turn over to humanized sequences, which will persist, I might say, indefinitely. And in that situation, they will keep working in the patient's circulation and on the tumor site.
Well, so you inject the cure and it stays there, continuing to treat the patient?
We hope that we will find a cure one day.
What does this mean to doctors now, would you say? Obviously, this is very early days for this therapy, but what is it telling cancer doctors?
So I think it will send a message that CAR therapy has a role in solid tumors and not just only in liquid tumors in hematologic malignancies. We know the CART is already very successful in hematologic malignancies, as we see the success story of CART-19. But this is an exciting start for the treatment of solid tumors with CART technology.
Now, you've got these CAR T cells, they have to be made for each patient. Is that a weakness?
I think the technology is developing very quickly. I don't think it's a weakness, because individualized treatment is also the strength of these CAR T cells. But we need only an apheresis, and the T cell of the patients is easy to collect. And after the technology is already available to very quickly generate these genetically engineered T cells.
So in a few words, how would you sum this all up? What to remember from your findings so far?
So we presented today that the CAR T cell technology will have effect on solid tumors, not just on liquid malignancies and hematologic malignancies. This is an early start, but I strongly believe that CAR technology and CAR treatment will win against solid tumors too.